Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So with that, I'd like to thank you for joining us today, and it is now my pleasure to introduce our speaker, James Brady from MaxCyte.
Hello, I'd like to start by welcoming all of you to our webinar. Today I'll be highlighting the advantages of MaxCyte's scalable electroporation-based transfection technology for expressing complex proteins and biologics, including multivalent antibodies, viral vectors, and multi-subunit membrane proteins. Before I start sharing data, I'd like to spend a few minutes to tell you about MaxCyte and the unique approach that we have taken to cellular transfection. Our technology was developed for clinical use, and therefore our focus has always been on providing a safe, non-viral approach to loading molecules into challenging cell types with high efficiency while maintaining high cell viability. MaxCyte cell loading technology is based on electroporation. We suspend cells in a physiologically balanced conductive buffer with relevant loading agents, and then apply a series of electrical pulses that create a temporary perturbation to the cell membrane, allowing molecules of almost any size to enter the cell very rapidly. Our motto is any molecule, any cell, any scale. In addition to plasmid DNA, we can efficiently load mRNA, siRNA, and even proteins into many different cell types. The technology can be used with a wide range of primary cells and cell lines, including insect cells. One of our technology's most unique features is scalability. We can transfect small numbers of cells, 500,000 cells up to a few 100 million cells, by static electroporation, which takes only a few seconds. Larger numbers of cells, between 500 million and 200 billion cells, can be transfected by flowing cells through the electroporation chamber. Because we apply the same series of electrical pulses for every transfection with a given cell type, we achieve very consistent results regardless of the scale. The MaxCyte delivery platform has been adopted by many pharmaceutical companies, biotechnology companies, academic institutions, and other organizations to develop a wide range of therapeutic modalities. We can generate modified cells for autologous and allogeneic cell therapies. We enable large-scale manufacturing of viral vectors and VLPs for vaccine and gene therapy applications. We can produce high-titer antibodies and other protein-based biologics in cell types that are relevant for bioproduction. And we can express complex drug targets such as ion channels and GPCRs in large numbers of cells for cell-based drug screening. In today's presentation, I'll share data related to all of the above application areas except for cell therapy. During my presentation, I'll highlight a number of key benefits of MaxCyte's delivery platform. With the MaxCyte system, you can work with the appropriate cell type, thus maximizing the relevance of your data. Users can produce a wide range of molecules, including complex multi-subunit proteins and viral vectors. High productivity stems from the high levels of transfection efficiency and viability that we can achieve, even with many cell types that are refractory to transfection with other methods. Our electroporation-based platform is much more consistent and scalable than chemical-based transfection methods, and due to the efficiency and scalability that we can achieve, we enable high transient productivity as well as more efficient production of stable pools and clones. Higher productivity, in turn, allows customers to dramatically shorten development timelines. In the next few slides, I want to dive a bit deeper into the details and key features of MaxCyte's scalable transfection technology. MaxCyte offers two different instruments for basic research, bioproduction, and high-throughput screening applications. The MaxCyte STX instrument can transfect as few as 500,000 cells or up to 2 × 10⁻⁻⁻ cells at one time. Smaller cell numbers, up to 700 million cells, are transfected in a few seconds. Larger cell numbers flow through the electroporation chamber. During electroporation, cells are suspended in MaxCyte's chemically defined electroporation buffer at high density, typically 100 to 200 million cells per mil. Thus, 2 × 10⁻⁻ cells can be suspended in 100 milliliters and processed through the STX in less than 30 minutes. Our VLX instrument can process 1 liter of concentrated cells, or up to 2 × 10⁻11 cells, in a single transfection run. I also want to point out that all transfections take place in sterile, single-use chambers called processing assemblies. We have different sizes of processing assemblies to accommodate different numbers of cells. Everything that comes in contact with the cell is single-use and disposable, so there's nothing to clean or sterilize in between uses. Finally, as I mentioned earlier, MaxCyte's technology was developed for use in the clinic. We have a master file with the FDA, and therefore our technology is ideally positioned for cGMP manufacturing applications. The MaxCyte technology is very simple to use. Users do not make any adjustments to the electrical parameters. They simply select a protocol optimized for their cell type from a drop-down menu. This is our current standard protocol list, which contains optimized protocols for over 80 cell types. Cells commonly used for bioproduction are highlighted in blue. If your cell of interest is not on this list, we also provide 10 optimization protocols ranked from low to high energy that should allow you to find easily conditions that provide good transfection efficiency and viability with almost any cell type. Here I'm showing you pictures and corresponding FACS data for cells that were transfected with GFP plasmid DNA to illustrate the high levels of transfection efficiency and viability that we can achieve via electroporation with plasmid. Note that the technology also provides highly efficient transfection with insect cells such as SF9s, thus eliminating the need for baculovirus. Now I'd like to share some data on scalable high-titer transient antibody production in CHO cells. At the top left side of this slide, you can see pictures of CHO-S cells that were electroporated with GFP plasmid DNA using the MaxCyte STX. 48 hours after transfection, almost all of the cells were expressing high levels of GFP, and viability was close to 100%. These levels of transfection efficiency and viability are typically much higher than can be achieved with other conventional transfection methods. To illustrate how high efficiency and viability translate to high productivity, we transfected CHO-S cells with a human IgG expression plasmid at small scale via static electroporation in an OC400 processing assembly, and at large scale via flow electroporation in a CL2 processing assembly. Both sets of cells were seeded into shake flasks at similar concentrations and cultured under identical conditions. You can see that the small-scale electroporation with 8 × 10⁷ cells and the large-scale electroporation with 1 × 10⁻⁻⁻ cells produce consistent antibody titers with comparable expression kinetics. Note that both sets of transfected cells yielded titers greater than 1.2 grams per liter. In this case, by using flow electroporation, more than 3 grams of protein could be produced in a 3-liter shake flask. I'd also like to point out that the high efficiency and high post-transfection viability afforded by the MaxCyte electroporation process can also yield benefits for stable pool and clone generation. Cells often recover more quickly from selection compared to other transfection methods, and often fewer clones need to be screened to generate a high-titer stable cell line. In addition, it's possible to parallel track stable clone generation while conducting analytical activities with transiently produced material. As depicted in this slide, a customer may transfect up to 2 × 10⁻⁻ cells on the STX for transient protein expression in a large shake flask or small bioreactor. A subset of those cells can be transferred to a smaller flask right after electroporation and subjected to selection to initiate the pathway for stable clone production. We have shown that in some cases the process for stable clone generation can be shortened to as few as 8 weeks. While cells are undergoing selection and expansion for clone generation, the transiently produced material can be used to generate functional or analytical data, including in vivo data from small animal studies. The data from the transient material can be used to determine if a particular candidate molecule is suitable for further development. Thus, the decision to advance or kill a candidate can be made while a clonal production cell line is still being developed. Now I'd like to share data on expressing conventional, or rather now that I've shared data on expressing conventional human IgGs in CHO cells, I'd like to discuss some of the more complex molecules that can be produced using MaxCyte's scalable electroporation technology. First, I'll show some examples of bispecific antibodies that were produced in CHO cells via transient expression. These data were provided to us by Dr. Matthias Pipe and members of his laboratory at Christian Albrechts University. They express bispecific antibodies in 2 formats, tandem scFv domains, also known as bispecific T-cell engaging or BiTE molecules, and a Tribody format. Both molecules have recognition domains for specific tumor cell antigens and immune cell antigens. The bispecific proteins were expressed by single plasmid or two plasmid co-transfection of CHO-S cells via static electroporation with the MaxCyte STX instrument. The graphs on the right show specific binding of the purified proteins to tumor cells expressing the relevant antigens. Here are functional data for one of the transiently expressed BiTE molecules. In this experiment, specific killing of lymphoma cells was induced by stimulation of non-activated T cells in the co-culture assay. Thus, MaxCyte's transfection technology allowed rapid, high-titer production of functional bispecific molecules in a cell type relevant for biomanufacturing. Next, I'd like to share data from another customer who used the MaxCyte STX to express multi-subunit ion channels via transient expression for a cell-based screening application. 3 plasmids encoding different subunits of voltage-gated calcium channels were co-transfected into adherent HEK cells, along with a 4th plasmid encoding an inward-rectifying potassium channel, which allowed the customer to modulate the cell's resting membrane potential by varying the external potassium concentration. The transfected cells were plated in 384-well plates immediately after electroporation, and the cells were assayed by measuring calcium flux 1 or 2 days post-transfection. This slide shows calcium flux data for 4 different calcium channels that were expressed on the surface of STX transfected cells. A robust calcium flux was observed in all 4 sets of cells. You can see that the calcium flux was eliminated following exposure to a specific channel inhibitor, demonstrating that the transiently transfected cells could be used to identify channel modulators in a screening assay. Thus, compared to the time and effort that are needed for generating stable cell lines, the MaxCyte transfection system provides a much simpler and faster approach to cell-based assay development, particularly with complex targets. Finally, I want to spend a few minutes to highlight the benefits of MaxCyte's scalable transfection technology for manufacturing viral vectors, VLPs, and vaccines. In this example, we were approached by a customer who wanted to produce a vaccine by expressing alphavirus-derived viral replicon particles in Vero cells. The customer's existing manufacturing process was based on co-transfecting the replicon RNA along with 2 plasmids that encoded helper proteins. They wanted to transition to a manufacturing process based on triple mRNA transfection. However, using other transfection methods, they were hindered by low transfection efficiency and mRNA degradation issues. MaxCyte provided a scalable manufacturing solution that allowed high transfection efficiency with both plasmid DNA and mRNA. Comparing MaxCyte transfection with the customer's existing transfection method, we achieved approximately 2-fold higher titers by co-transfecting mRNA with plasmids, and we achieved 10-fold higher titers by co-transfecting all of the VRP components as mRNAs. The last set of data that I'd like to share relates to manufacturing lentiviral gene therapy vectors. This slide highlights some of the challenges associated with traditional approaches to producing lentivirus. First, stable producer cell lines are challenging to generate due to toxicity of viral proteins. Therefore, lentivirus is typically generated by co-transfecting 3 to 5 different component plasmids into unmodified cells. Adherent hex cells are most commonly used for lentivector production because those cells are most amenable to reagent-based transfection methods such as calcium phosphate or PEI. However, large-scale transient expression in adherent cells presents a number of logistical challenges, particularly when trying to meet the dosing requirements for a clinical study. In addition, titer often drops substantially during process scale-up when using calcium phosphate transfection. And then finally, due to the amount of time that it takes for plasmid DNA to enter the cell and migrate to the nucleus following calphate or PEI transfection, the final vector harvest may not take place until 3 or 4 days after transfection. MaxCyte's scalable electroporation technology offers several key advantages for lentivector manufacturing. First, it yields high transfection efficiency with both adherent and suspension-adapted cells. Transitioning to suspension-adapted cells allows for much simpler manufacturing scalability, and titer is also maintained during process scale-up with MaxCyte's technology. Finally, because DNA enters the cell and migrates to the nucleus more quickly with electroporation, production runs can often be shortened by at least 1 day compared to traditional lentivector manufacturing protocols. Here are data from a lenti-vector manufacturing process that we developed in collaboration with Dr. Ken Cornetta's laboratory at Indiana University. Suspension-adapted cells cultured in wave bioreactors were transfected via flow electroporation with 4 HIV-based lentiviral vector component plasmids. Titers obtained via MaxCyte's electroporation method were comparable to or even greater than titers achieved by calcium phosphate transfection with adherent cells. The table shows titer data from 3 independent production runs that were conducted in a GMP suite. Note the consistency of titers, approximately 1 × 10⁸ transducing units per mil, achieved in all 3 runs. In conclusion, I'd like to highlight once again the key benefits of MaxCyte's scalable cell delivery platform. With a MaxCyte system, you can work with the appropriate cell type. Users can produce a wide range of molecules. High productivity stems from high transfection efficiency and high viability. MaxCyte's electroporation-based platform is much more consistent and scalable than chemical-based transfection methods. Efficiency and scalability enable high transient productivity, as well as more efficient production of stable pools and clones. Higher productivity in turn allows customers to dramatically shorten their development timelines. So at this point, I'd like to thank all of you for attending our webinar, and I'd be happy to answer any questions in the remaining minutes that we have left.
Thanks, James. So the first question we have is, for multi-plasmid transfections, is it important to optimize the relative plasmid ratios?
Yeah, so that's an interesting question. Certainly optimizing the ratio can make significant differences if you're, say, producing a viral vector. As I mentioned earlier, some of the components can be toxic to the cell, so minimizing the ratio of those toxic plasmids, maximizing the ratio of the other plasmids can improve viability as well as titers. If you look at the data I shared earlier on the multi-subunit calcium channels, in that case the customer did not do any plasmid optimization. They simply used a 1:1:1 ratio of all the plasmids. So whereas plasmid ratio optimization can certainly help, extensive optimization is not always necessary.
Okay. And what are the most critical variables for ensuring good transfection efficiency and productivity with your technology?
So because our technology is based on electroporation, we deliver the same series of electrical pulses every time you hit that button. So there's really very little variability in our process. The 2 biggest variables in our process are the cells and whatever you're putting into the cells. So it is important to have cells with high viability. They should be actively dividing. Early passage cells are preferable. And then, of course, if you're using DNA, high-quality DNA is important. DNA can be prepared using any type of conventional preparation methods. We recommend putting the DNA in water rather than TE. And, of course, the DNA should be assayed on a gel to make sure it's intact and that it's not nicked or linearized. But if you have healthy cells and good quality DNA, those are the most important variables for getting good transfection results.
Okay, and besides efficiency and viability, what other variables influence productivity with the transfected cells?
So certainly there are a number of variables relating to the culture process that are going to influence your productivity, the base media that you use, the feed additives that you put into the cell culture. Doing a temperature shift in the case of CHO cells down to 32 degrees can certainly improve productivity. And then if you put a little bit more effort into monitoring metabolites and waste products and adjusting the pH, all of those activities fall under the rubric of process development. If you put some effort into process development, you can really achieve very high titers via transient expression with our technology. And I should also mention that when customers acquire the MaxCyte technology, they also acquire access to our scientific team who will work very closely with customers to help them to optimize all of those different process variables.
Okay, and it looks like we have time for one more question. Does MaxCyte require special media or culture conditions?
No, so the other advantage of our technology is that during electroporation, cells are suspended in our buffer, which is a physiological salt solution. We use 1 buffer for all cell types. Then after electroporation or prior to electroporation, cells can be cultured in any type of culture media, any type of culture vessel. So a lot of customers like to work with our technology because they can use their specific cells, their specific media, and their specific feed additives.
Well, thank you, James, and thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts covering many aspects of bioprocessing. Look for those announcements in your inbox. Thank you for attending this BioProcess International Ask the Expert webcast. Have a great day.